present you a bit more about more complex synthetic circuits, mainly focusing on how to implement different logic circuits where you can have as input different signal to take them into consideration. And so here is just like a small example about two ideas about how you could use these more complex systems. And here is one where you will have as input some nitrogen and the presence of some plant beneficial bacteria, and you will have as output, when those two is present, the development of different root architecture. And so this is just a rough example about how you could use this type of system. And here on the side is a metabolic pathway where you will want to induce the production of a specific metabolite when you have the presence of light, for example, and the presence of the primary metabolite, which is there. So just some example here. So the general topic that I will go on in this presentation is the structure of complex circuits. So that they are composed of input, sensing, signal processing, and output. And then I will go over different tools to implement these logic circuits. And then I will give a short example about how to design different circuits to be able to optimize the system that you want to implement. And how to do so. So just it's not touching anymore. Okay. So for synthetic signaling circuits, so we, as Nicola presented, we can have a lot of different input for the system. So there could be environmental input, such as stress, abiotic or biotic stress. We can also have intracellular molecules, such as hormone, or we can also have any other promoters that are like tissue-specific or specific to any cellular states in the cell. And then we'll have some sensing systems that could be some specific promoters that will then lead to the processing part, where we will process those different information and corresponding to some logic circuits. And Adil presented to you yesterday already some very nice NOR circuits. So so this type of system are quite interesting to be able to process different inputs. And then we can have a lot of different outputs that could be some reporters, so some fluorescent protein or other. That could be the production of some metabolic pathways, such as expression of an enzyme. And it can also be the switch to a new cellular state or to any other phenotype of interest. And Alex gave a good example here also about development of lateral root from the system of Gen with this buffer gate. So I would just go over these different tools for signal processing. So it's not an exhaustive list, it's just kind of some example of the ones that are quite highly used, but there is a lot of other ones that have been developed or could be developed. So the first one is using transcription factors. So it could be transcription activator or repressor. The second one is using CRISPR-Cas9, where you produce some sgRNA, which could lead to repression or activation of the output gene. And the third one is different post-translational tools that I will present, such as protease or some split enzyme system. And the final one is integrase that I went over a bit yesterday, and I will show you how we could use it and present you a bit of the difference on why using one or the other. So starting with transcription factor. So as Nicola presented a bit, is that you have most of the time some binding domain, and then you could have some some transcriptional activator, transcriptional activator that will be fused to the binding domain. So then when you have the expression of this protein, it will bind to this promoter that is composed to some multiple binding sites, and then a minimal core promoter. and that will lead to the expression of your output gene. And there is a lot of different activator domains that have been used, such as VP16, that is one of the ones that have been highly used, or you can also have some other one like ERF2. Yeah, so this gate is similar to the buffer gate that Alex presented, that you will, when you have the input that is present, you will have the expression of the transcriptional activator, which will lead to the expression of the output gene. So another one is a transcriptional repressor. So here the design is a bit different. When you don't have the presence of this transcriptional repressor, you will have the expression of the output gene. So this promoter, this synthetic promoter, is composed of a constitutive promoter, and then you will have some binding sites where the transcriptional repressor will bind to and will stop the transcription. And this could be done only by the fact that you will bind to the promoter, so then it will stop the expression, or you could also have the binding domain fused to some repressor domain that could be TBL or ERF4 or some other ones. So here you will have the, when you have the presence of the input, you will not have expression anymore. And what is kind of nice with this system is that you can also put them together. And here is an example of using two transcription repressor to implement this NOR logic gate. And so you have, you just put in your synthetic promoter binding site for the respective two transcriptional repressor. And so you will, if you have those, only when you have the two inputs that is present, you will not have the output anymore. Okay, and here is an example that is from Jennifer Brophy, and here you have a bit more complex system that is N implied B, and it's kind of an example about how you implement it in bit-obsessed Italiana, for example, and having as input signal some different promoters that are expressed only in some specific tissues. So here is an example with SMB, pin4 and pin4 as the two inputs. So you can see here just a very rough explanation where they are expressed in which tissues. And so here you have a transcriptional activator and a transcriptional repressor. And so if you have the input A that is present, you will have expression, and if you have the input B, you will not have expression. So that gives you normally this type of expression pattern. And here you can see that this is what has been obtained into Arabidopsis, which is quite nice and fit quite well to the prediction. And you can see from those very simple diagrams that I show you, in fact, the design is quite more complex. You have here the synthetic promoter with the different binding sites, the output gene here, and you also need to express the transcriptional activator, transcriptional repressor. So here for the transcriptional activator, it's this DNA binding domain and this activator. And here for the pin4, you have the DNA binding domain here with a nuclear localization signal tag. Yeah, so this is quite nice to see from a very rough circuit design principle to really actually how do you place those different components together in one piece of DNA. And you can see here that you, first have one gene, then the second one, and then the third one after the other. Yeah, and I hope I'm not saying anything wrong, Jen, so yeah. Okay, so then the second example is a CRISPR interference to implement logic circuits. And here it's been presented quite well yesterday by Adil, but I would just go over it a bit more. And so here's the principle. So you have a lot of different CRISPR system to implement logic circuits, but yeah, here I'm just focusing on the nice one that Adil developed. So here you have the inputs that will lead to the expression of sgRNA that is kind of the binding domain of the system that will allow the binding of the DCAS to the synthetic promoter, and then it will lead to the repression of the expression of your promoter. And so you will not have expression anymore. And so here, what is quite kind of nice is that what they've done is that you implement different NOR gates. So NOR gate is that you have you put two different DNA to have the sgRNA to be able to bind and repress. So then you have expression only when you have when you have none of the input that is present. And you can also have as an output of this first NOR gate having the expression of the other sgRNA that will be the input of the second NOR gate. So by layering those gates, you can implement more complex systems. So this is what Adil have done, and you can see from a very simple design here to more complex design and all the different components that you need to have. So here is with constitutive expression of the different sgRNA, and then you have this, all of these circuits with expression also of the DCAS9, and what is the output here. Okay, so the third one that I wanted to present is an post-translational regulation tool. So what is quite interesting with post-translational regulation is that it can really allow to have some quite quick changes. In comparison to transcriptional system where you need to have expression of your genes that can take a bit of time. So here is using protease. So yeah, here's the input mediates the expression of the protease, which will cleave to protein at the protease cleaving site here. And what the output that they use in the system is ROSEA1, which is a transcriptional factor which induces accumulation of anthocyanins. So as you can see here, it leads in the leaf. So this is a bent leaf to the expression to a bit more like reddish color. Yeah, it's not that great in picture, but they have some very nice picture where you can see that the whole leaf become red. So here, first you have a fusion between this ROSE1 and a gray protein that blocks the activation of the output protein. And those gray protein are viral protein, and they have different viral proteins that allow to block the activation of this transcription factor. So here, when you have the production of the protease, you will have cleavage, and then you will be able to have an active ROSE1 in the system. And also what they've done is to implement some AND gates, where here they have the production of two different proteases that recognize two different cleavage sites, and you have two different viral proteins that block the activation of this ROSE1 transcription factor. So you need to cleave both of them to have the protein to be active and to have your output. So this is quite a nice system as well, and you can implement more complex systems and the AND gate as well. And another one that is quite interesting is split protein with interacting domains. So this has been implemented in quite different system, and here I just show you one example. For this one, you have so basically in dark brown, it corresponds to the binding domain, and in dark brown, it corresponds to transcriptional activator, so to the activation domain. So you need to have the expression of both to be able to have the expression of your output gene, so which corresponds to an AND gate. And in this case, you have different hormones that will lead to the expression of this different protein. And the way this works is that you have those interacting domains that will make the protein come together and form this complex. So here is a full system that has been developed, and so those interacting domains are called HD here, and that you get the final expression of luciferase when you have the presence of those two proteins that come together. And those interacting domains have been used quite a lot in different systems, and they allow to implement this quite easily, this AND gates, and those work quite well. Okay, on to the last one. Sorry, it didn't appear at a good time. Is integrase. So I would say one of the big differences of the integrase is that in comparison to all the previous circuits, is that they mediate irreversible DNA switch, meaning that when the input is considered as on in those logic circuits when the input has been present, even if the input is not present anymore. So that is very nice to allow an irreversible switch and to allow, for example, cell lineage tracing, and to allow switch in a specific tissue, but it's not very nice to be able to respond to some quick change of environmental signals, because when you switch, forever, basically, in most of the systems that have been developed. You can reverse the switch, but it's quite more complicated. So it has this advantage, but also this drawback. So as I presented yesterday, the integrase recognize two different sites, and then you can have inversion of the DNA between the two sites when the sites are in opposite orientation, and you can have excision of the DNA between the two sites when they are in the same orientation. And we have different integrases that recognize different sites and that are orthogonal from each other. So this system can allow cell lineage tracing, such as what I presented yesterday, when you have the integrase that is under the control of a lateral root initiation promoter, and then you can switch to the expression of a different fluorescent protein in the lateral root. And another system that have been recently developed is implementing some more complex logic circuits, such as here you have 4 different integrases, which are Bixby1, FIS31, integrase 9, and integrase 13, and you have the different inputs that lead to the expression of those integrases. And here is the implementation of a quite complex logic circuits, which is NOR, AB, and XOR, CD. And basically how that works is that when you have the input A or the input B, you will have excision of these genes, because those two blue pair of integrase sites are in the same orientation, so you will have excision of the whole cassette. But then if you have the input C, so this corresponds to the NOR AB, and if you have the input C or the input D, you will have inversion of the DNA, but then if you invert it one, you will have expression. If you invert it twice, you will not have expression anymore. So this corresponds to the XOR CD. So this is how they implement quite nicely a quite complex logic circuits, which is quite fun, and to see if that can be useful for any of the systems that you would want to implement. Okay, so this is quite a novel view on all those different systems. And what I feel is quite interesting for you here, as you will think about designing some genetic circuits, is that which system do you need to design to be able to optimize your final circuits? Because those circuits can be quite complex, and you will want to have some parts that you will want to design and test and see and learn from this, and then combine them to build some more complex ones. And you also need controls to make sure that the system, that you can compare it to a baseline. And so I'm taking as an example some of the results from Jen paper, and this is, so the first example is using this activator, and what she've done is to test first a different number of binding sites, so from 6 to 1, testing what would be the design that will work the best, and having as control just this minimal promoter and the GFP. So to be able to compare it with this system. And what is shown here is that having 6 of those binding sites is what will work the best. So this is what is selected later on to implement some more complex system. And then to test those more complex circuits afterwards, I think it's going to be the A and imply B. What has been done is to test it in bands and to have first one where you only have the output. So you will have GFP and M-Cherry construct. So M-Cherry used to do calibration. And GFP is the one that will have this synthetic promoter in front with the different DNA binding sites. And then you will have other ones with the output and also different inputs. So either one input corresponding to the transcription activator or transcription repressor, constitutively expressed. So you can test when you have the input A, the input B, or when you have both of them. And you can compare that. So this will correspond to those 3 different bar graphs and the expression of the output genes that you have here. And so here what have been tested is different transcriptional activator and transcriptional repressor for the N imply B gate. So you have as transcriptional activator, you have either using the VP16 activator domain or using the ERF2 activator domain. And then for the transcriptional repressor, either not using any transcription, any repressor domain or using the ERF4 repression domain. And then testing those different on which gate would work the best. It ended up being this one that works the best using ERF2 and ERF4. And as you can see, as it's been tested before, that 6 binding site works the best for the translation activator. This is what is used here in all of them. So it's quite a good example about how you could have an idea about what you want to do and which different circuits you might want to design to be able to test and have ones that will work well. Yeah, and that's it. So I presented you some different examples about how you can process multiple inputs and then how you can test it and have a design that will work at the end. And yeah, if you have any question, feel free to ask.